Ken.
Great. Welcome back. I'm Ian Thompson, and wanted just a little bit of housekeeping before we turn it over. This is our schedule for today. Just remember, this may be an open meeting, so speak accordingly. Seth and Dan will start off our bladder organ site, followed by Drs. Lynn and Quinn, and then we'll have a brief wrap up and announcements at the very end. I want you to, for those of you who are not there, not joining us yesterday, just remember that a lot of functions that are made possible by the GU committee are made possible by the Hope Foundation, which supports, for example, the Young Investigator Training Course that we have benefited enormously from, as has many of our committee members. This is the QR code. If you take your phone and you hand it up to the thing, you can snap a picture, or you can go to the hopefoundation.org. And if we as a group help the Hope Foundation as a group, it will come back and help us many times over. Yesterday, we passed the baton from Dr. Vogelzang as vice chair of the group, and to Dr. Petrolak. Welcome, Dr. Petrolak. We're very glad that you're filling his shoes, and we hope to have a in-person going away party for, even though he's not going away, Dr. Vogelzang. And one more time, next go around, Dr. Flake will be joining Dr. Lerner as an organ site chair for running our meetings. And finally, one last reminder, in order to receive an email about the GU committee news, you have to be a member. So some of you on this call may not currently be members. I'm not sure if that's true or not, but if you're not a member, protocols@swog.org, or you can email any of us and we can put you in touch with folks to make that happen. And finally, let's all stay on time. And with that, I'll stop my share, and Seth, you're on.
Super. I'm Seth Lerner. Welcome, everybody. Just want to, again, give my personal thanks to Dan Petrolak for really all the great work that he's done over the years, and congratulations on being promoted to vice chair. And welcome to Tom Flake. Tom's been a stalwart in the bladder committee and obviously chaired 1314, has done some fantastic work, and really thrilled to get to work with you. Tom, we have monthly bladder calls, fourth Wednesday of every month, 4 to 5 o'clock Central Standard Time. Taj, please email Taj if you want to be on that list. It's a big tent. We welcome everybody, and we'll get a move on. This is our agenda. We're going to spend a little bit of time on our 2 most recently activated trials, and then we'll give you an update. Yeah.
Your slides for us are not advancing.
Oh, come on.
Sorry. Maybe you need to stop sharing and try again.
Jesus. Okay.
Seth, do you have a dual screen or a single screen?
Single screen.
And do you have the option for presenter mode and swapping your screens?
I have no idea. You're talking about within
Within PowerPoint.
Okay, so let me get out of this. Okay, and now what do you want me to do?
Right, Seth, try the non-presenter mode. Just, click on one slide and just make it as big as you can, and then share that.
Oh, I see what you want me to do. Okay. I don't know why that's doing this.
You're showing us what you're looking at on your laptop if you were presenting it on a projector.
Oh, yeah. Okay, hang on.
Might want to turn it up more.
How about that?
You got to share the screen.
Yep, hang on. How's that?
Perfect.
You got it.
Hallelujah.
You want me to start over from the beginning? No. All right, I just ate into about 10 minutes of Queen and stuff. My apologies to those of you who have slides on the back of this slide deck that we may not get to you. Anyway, okay, Sarmad Sadeghi, who's been patiently waiting. Just tell me when to advance the slides, big guy. Go ahead.
All right. Hi, everybody. Thank you for giving me the time to bring you up to speed on SWOG 1937. This is, by now, I think everybody knows about this study, and today I'm going to make an appeal to everyone to please activate the study at your sites, and I'll go over some of the information that we have. So this is a phase 3 randomized trial for previously treated metastatic urothelial carcinoma patients. We want patients that are post-immunotherapy or immunotherapy ineligible. The study has 3 arms. 2 of them have eribulin. One is eribulin monotherapy. The other one is eribulin plus gemcitabine. And the control arm is what we are very used to. It's docetaxel, paclitaxel, or gemcitabine. Next slide, please. So this study was designed as a late study in the course of treatment, maybe even 4th line, but with changes that are happening, the landscape is changing very quickly. And I think some of the, some sites that have been slow to look at the study and open it may have done so because of that, perception that it is not very relevant. I want to make the case today that it is very relevant, and I want to draw your attention to some of the recent changes that we had just over the past few months. July of 2021, and enfortumab vedotin. Now you can give it to patients that have not received really platinum-based chemotherapy, and if they have received therapy, they're good to get it.
If you ever need for your journal.
And then we have the approval of Nivolumab in adjuvant setting. These are patients that get cystectomy, high-risk disease, they get Nivolumab. And if they progress, then suddenly they're post-immunotherapy, depending on what they have received before cystectomy in the adjuvant setting. They may already have had cisplatin-based chemotherapy. So suddenly in metastatic setting, 2 of the major lines of treatment are exhausted. And in frontline, the indication for pembrolizumab is now a full approval for patients that are not platinum eligible. They can get pembrolizumab in frontline treatment. And by definition, these are patients that are not platinum eligible. So all they have left is enfortumab, and then they are in need of treatment. Next slide, please. So this is what this landscape looks like. And I have put, 2 paths here. One is metastatic path, and the other one is resectable urothelial carcinoma. And you can see SWOG 1937 is now, in 3rd line. It's an option in the 3rd line. And we expect some additional changes, and enfortumab plus pembrolizumab may become an option, and suddenly you could, see this move even, a line earlier. So it is a very relevant study in our view, and I would like to ask everyone to please, when you go back, we're not traveling for this meeting, but when you go back to your routines of every day, please look at this study very, very carefully and consider opening it at your site. Next slide, please. So we still have significant unmet need in this space. We have sacituzumab govitecan with accelerated approval, but I want to point out that the phase 3 is comparing sacituzumab to docetaxel and paclitaxel. So it would not be wrong for anyone to say that if we are comparing something to docetaxel, paclitaxel, we may as well randomize these patients on SWOG 1937. We have 2 out of 3 chances of getting the experimental agent eribulin, which in phase 2 studies has response rates between 35 to 50%, and sacituzumab is 27% response rate. So I want to make the case that we should not feel that we have to do sacituzumab for every patient. The equipoise exists for us to consider SWOG 1937. And then, as I pointed out, as treatment lines are collapsing, enfortumab vedotin combined with pembrolizumab, we may get there faster, with treatment options for our patients. And then finally, this is a very flexible study. The performance status requirements are for real life patients in this situation. So patients can easily get on this study. It's not sensitive to the number of lines of treatment, what has happened before and what has happened after. As long as patients have had immunotherapy and are previously treated, they are considered eligible. We are also going through an amendment process. Some of the criteria are getting even more relaxed. We had creatinine clearance of 50, and after 2 or 3 patients who could not be screened for this study, we are reducing that to 30 cc's per minute. Next slide, please. So this is the setup. As I said, 3 arms. 2 of them will give the experimental agent to the patient, and the 3rd arm is the control arm, docetaxel, paclitaxel, and gemcitabine. Criteria for inclusion are shown on the left. Next slide. I'm not going to go too much into this slide. We have seen this multiple times. This is 465 patients, 155 in each arm. And these are stratified based on Belmont risk groups and whether or not they have received immunotherapy or they are immunotherapy ineligible. And the endpoints are the usual suspects: overall survival as the primary, and PFS, objective response rate, and duration of response, and as other secondary endpoints. Next slide. We activated this study in February of this year. We opened it at USC in March. We have 2 patients enrolled on the study as of this week. At USC, I think there are other patients that are going on in screening, but a lot of very productive SWOG sites have not opened the study. What I hear is that the COVID impact and the coordinator turnaround have been major contributors to this slow start. And we are sending out a survey to SWOG investigators at various sites to ask them what the status is, whether they intend to open the study or not, and what are the impediments. I'd like to ask every one of you to please respond to that survey and let us know how we can help you overcome some of these impediments and get the study open. As the landscape is changing, this study is becoming more and more relevant. Next slide, please. I'd like to thank everybody that has participated during this, process of getting this study off the ground. I'd like to especially thank Rick Banks, who's been always there helping me with the all sorts of issues. He's my go-to person for advice, and he's going to help us get this study activated at more and more sites. Seth had in the agenda that I was going to talk a few minutes about FRNB2 study, which we recently presented at ESMO. I don't think we have time. If there is time, I'm happy to discuss it, but we can do that offline, I think. So
great. Yeah, Dan Petrolak, you want to say?
A couple of things. Firstly, is Alliance and ECOG, are they open at this point or not?
They're going through the same difficulties that most of our sites are going through. Scott Tagawa, I think he's almost ready to open it. He's been very, very helpful in all of this, but I'm not aware of other sites.
So what I would it's just unfortunate that the ASCO GU deadline just passed this week, because this would have been a perfect trials in progress abstract to have presented. Perhaps we could do that for ASCO. You could prepare a trials in progress poster.
It's an excellent idea.
And this way we can let people know this is available. You can get this out to the general population that the trial is open. And again, I think I agree with your survey. I think contact individual sites and finding out what their challenges are is the right thing to do.
So like we discussed yesterday, I think this got trapped in that purgatory of clinical research professionals being in rare supply, especially regulatory CRCs. And I know of about 80% of the CTOs around the nation are needing regulatory CRCs, and that's why the activation timelines have slowed down markedly this year.
So I think this is a big problem right across the board. And I realize this because I'm trying to get some of my studies that are in the early clinical trials network done and SWOG and stuff. And many people who are friends and investigators of places are just saying, look, we don't have the staff to run these trials. The one point I'll make to you, Lucky, and I think this needs to be communicated more broadly, the NCI has no idea that this is a problem. They seem to be acting like COVID didn't occur. And in several discussions with good people at NCI, I'm being told, okay, get your trial moving. And I'm saying, well, listen, this is the response that we're getting. And this is why people are not opening, whereas in the past they would have opened everything. Hopefully that's going to change.
Well, actually, David, you're correct. In fact, this came up last week during the NCI Cancer Center directors meeting. So even Ned Sharpless is aware now that this is happening across the nation.
Is that why there's so many suspended sites?
Yeah.
Probably.
Yeah, so I mean, there's if you look at this trial, there's just dozens of suspended sites. And so there's hard data there on what's happening. That might be helpful on the
And I
NCI
think
side.
each site is a different issue. It could either be the regulatory where they can't get it through regulatory, or some sites like mine, we are on a voluntary hold since June for new accruals because of the lack of people to put data into the databases.
Yeah.
So we had these really massive data backups because we had nobody to put the data in.
So Lucky, at the Cancer Center directors meeting, did Ned, because it's my impression as well. So at the BSA, it has not come up. Which surprised me, and probably that's my fault for not bringing it up. But was there anything? Did they say they were going to be taking some steps to address it?
No, the discussion happened during the presentation of the CDER, the FDA CDER director, I forgot her name, gave a presentation, and that's where it came up. The cancer center directors, most of us expressed our concern about the attrition of clinical research professionals across the CTOs, and we were asking the FDA to help us out, because a lot of the attrition is because not just of staff but faculty too, because of mostly because of the demand, especially of industry, to get data by this afternoon. These sort of pressures on the staff that contribute to burnout. There was no action item, Ian, if that's what.
Yeah, that's what the question was, but,
Yeah.
but I'll follow up on it.
Yeah, if I may, that I actually brought this to the attention of the woman who heads up the Clinical and Translational Advisory Committee, which is an advisory committee to Ned Sharpless, and I was asking for some data. And unfortunately, the data that they have show that that accrual overall globally to NCTN trials is actually held pretty well, but there's obviously a disconnect there. So there may be an opportunity. Our next meeting is next month, and I'll circle back and see if this can get on the agenda, at least for some initial discussion and input. But if it's okay with you, let's move on to the next trials. Very helpful discussion. So Guru will present the recently activated S2011.
Thanks, Seth. Yeah, exciting to get this trial going finally. So this is the SWOG 2011 randomized phase 2 trial evaluating gemcitabine carboplatin combined with avelumab as neoadjuvant therapy for cisplatin ineligible muscle invasive urothelial carcinoma. Of course, Alliance and ECOG and NRG are also expected to participate in it. And so it just activated last week, and I hope we will have participants. Okay, next slide. So this is the design slide, just a single slide I'm showing you on this study. So it's a randomized phase 2 trial. It's inclusive in design, the eligibility, so muscle invasive, upper tract urothelial, as well as bladder, cisplatin ineligible, and a predominant urothelial component and no small cell component. And to see the stratification factors here, we're not stratifying for PD-L1, we're stratifying for clinical stage, performance status, and renal function. So that's a very desirable feature because all these other trials going on, we have to wait for central PD-L1 testing and delay. There's a lot of delay in getting treatment going. And we reason that the PD-L1 should get balanced out in a trial of this size. So upfront surgery versus 4 cycles of or about 12 weeks gem carbo plus avelumab. Avelumab is every 2 weeks, and gem carbo is as usual, day 1, day 8 of every 3 weeks per cycle. And then the radical cystectomy, urethrectomy, or nephrouretrectomy. So one of these surgeries based on the location. And the other thing I wanted to highlight is that in the adjuvant side of things, we are allowing adjuvant nivolumab per investigator discretion, of course, in both arms actually. So depending on whether you feel a patient needs nivolumab, that is allowed, which is also nice because the other ongoing neoadjuvant trials actually do not allow adjuvant nivolumab. So that's also a nice aspect to this design. So it's a very inclusive study and incorporates the current standard of care. So the primary endpoint is PAT-CR with secondary endpoints of EFS, OS, and correlative endpoints, blood and urine correlative endpoints, 178 evaluable patients. And we're seeking an improvement in PAT-CR from 15% in the standard arm to 35% in the experimental arm. So I'm hoping people participate, and I think this is an attractive design in my opinion, and it complements the other ongoing neoadjuvant trials. Any questions?
Yeah, I had no questions. I would just like to echo that this should be a relatively easy trial to accrue to. It's a disease state that definitely represents an unmet needs. Radical cystectomy or surgery is the standard of care, and here's an opportunity to test a rationally designed combination chemo-IO, and it's a relatively modest group of patients with a hard endpoint of PAT-CR, lots of cool correlatives. So I hope everybody can get on board. It's and we've got champions in the other cooperative groups, I believe. Guru, is that correct?
That's correct, Seth. So we have champions in all the other cooperative groups. There's Rana McKay, there's Suzanne Cole, and Shilpa Gupta actually is from Alliance. So we have in all the groups.
Great, thanks. Okay, so I'd like to introduce to people who are well known to us, Lucky, Lara, and Kathy Albane, to just talk very briefly about the SWOG Clinical Trials Partnership. And I think Lucky's going to say a few words first.
Yeah, really a few words. You all are aware of our frustration with having fairly limited bandwidth when doing clinical trials within the GU committee, and for that matter across SWOG. And we sort of designed these concepts, and we have to squeeze it into the GU steering committee and get NCI blessing for our concepts, even though we have limitless other bright ideas and really highly impactful concepts. So when there was an opportunity to develop the SWOG Clinical Trials Partnership, this was a way for us to widen the bandwidth of our activities and be able to do high impact studies without necessarily limiting ourselves to the NCI mechanism. And so Dr. Kathy Albane, who's well known to you, was appointed our vice chair for clinical trials partnerships. And she has a very brief slide set here that I think summarizes what the value of these of this effort is and how it now directly applies to a concept that is being advanced through the bladder cancer organ site. So Kathy?
Thank you, Lucky. Thank you, Seth. Thank you, Ian. And it's good to see all of you. Next slide, please. So as Lucky just mentioned, SWOG Clinical Trials Partnership, CTP, establishes and administers preferably preferred partnerships between SWOG and industry, where we allow them to collaborate directly with us as a preferred partner, study platforms across all phases of clinical research. And this partnership is to facilitate effective, rather efficient and effective testing of new agents and combinations across disease sites or broadly within a disease. It flows through the CTP channels that are dedicated to study design, review, approval, contracting, and is done concurrently with industry, and happily taps the same outstanding operations, statistics, and data management personnel as we are used to in our NCI-funded studies, but uses non-federal dollars for the CTP efforts. Next slide. So this explains it perhaps in an easy way to understand. To the left on the slide are the NCTN-run federally funded SWOG trials that utilize industry support for drug costs, distribution, other support. And we've been doing these types of efforts under previously CTI and then now CTP. This continues, but these are for our federally funded trials and are not the topic of this presentation. To the right, there are the CTP-run, rigorously scientifically relevant industry-supported trials that are supported 100% by industry. Down to the left, we can still do single SWOG committee-initiated studies in the CTP mechanism. We had done those as clinical trial initiatives before in SWOG, but these are not to be viewed as one-off trials, looking for a new place in the market for a certain drug, so to speak. And those are still important trials, but for us, we want to select companies to work with where we could potentially see a more complex or thorough partnership evolve over time. And that would be a preferred partnership, the Preferred Partnerships or PPP program within CTP, which is the new mechanism that Lucky discussed. It's for pipeline access or complex multi-arm platforms with joint scientific development and governance from the get-go, and utilizes our dedicated CTP infrastructure. And importantly, these do not overlap or compete with active federally funded trials. Next slide. The CTP process involves parallel study design jointly with industry, scientifically, budgets and contract negotiations, and protocol development and study build. Shown on the next slide in much more detailed fashion, which I'm not going to review, but just pointing out, if you look at the purple boxes, the second row, which is the scientific clinical aspect of our study development process. As you go along by the first red arrow, that's where you would normally send a project to the steering committee. But since these are not NCI funded trials, we will have our own executive scientific review, so-called triage, to approve the capsule. And then going along the usual processes we're used to with the PRC at the Statistical Center, rapid review, all the way to the second red arrow there, which is the CTP executive review body of the entire protocol instead of going back to NCI. So this will be very familiar to all of your research teams, simply that it is not using NCI scientific review process or funding. Next slide. So right now we have a very active menu already of industry collaborators where we are developing pipeline, platform, or subtype driven trials. For example, Dr. Blanky announced at the plenary session yesterday that with Novartis, we now have a preferred partnership program agreement signed for many assets in their pipeline across disease sites. So GU committee, stay tuned. There will be some conference calls set up of a lot of great opportunities. Their pipeline is very deep, and hopefully there'll be some projects there. But next slide, we do have one going on now with Genentech, which will be covered next by Josh Meeks. This is a very exciting screening platform design. And I will stop here and either take questions or let Josh proceed. Your call, Seth.
Yeah, if
Thank
it's
you for the
okay.
chance to talk.
Yeah, why don't we press on? I just want to say publicly to Kathy and Lucky and the whole CTP team, we approached them about, I don't know, seems like 12, 18 months ago when I think that both of us were just getting started. It's exciting to see how the program has evolved, and you're about to hear about a topic that I think you're familiar with, but we've now made a lot of progress. So Josh is going to walk us through the trial that Kathy just introduced.
Great. Why don't you go ahead and advance to the next slide? So again, just like Seth mentioned, I want to thank everyone on our side, from the GU committee. Seth is my co-PI with this, and then of course, Kathy, Tom, David, and Peter, and then Dr. Laura Albane from the CTP group. Next slide. So this was the overall strategy that everyone's kind of seen this. This work really began, interestingly, with many of the investigators from the GU committee, from the Cancer Genome Atlas, with the thought that in bladder cancer, we could begin to look at the gene expression profile of each tumor through tumor subtyping. And then that in itself could lead to an RNA-based biomarker. And so the overall concept is that you could, again, use the tumor tissue, build in integrated biomarkers. That's okay, you can advance that. And then from that, if you look at pathologic response as a response to that novel therapeutics, we can then make some very early assessments of, is this working or not? And a futility analysis to decide if this is the best therapy for this tumor subtype. Next slide. And so the updates, I just want to give a brief update of where we stand. A lot of work under, a lot of water under the bridge. So the first, and then as Kathy mentioned, is that we have a partner. So Genentech is our partner for the bladder subtyping CTP, and we really couldn't do this without them. And of course, we really acknowledge Michelle Brockman, who just has been a great partner for this, as well as many of our other trials that we've worked on here. Second, I think we're closer to developing a schema that will be ready to begin. And I borrowed this from Kathy Tengen because she said this best in an email, that really this is, even though this is a trial, it really is 3 trials, and it is a bigger undertaking. So next slide, you can see the schema. And so just to walk you through this briefly, patients come in, they're muscle invasive bladder cancer. They would have their tumor subtyped, and then they'd be put into one of 3 groups. And the reason even though there's been a lot of discussion about how many subtypes there are, we all sort of agree that there's on one extreme luminals, on the other there are basals, and there are some that don't really fit in there, and we put them into a non-luminal, non-basal group. So the luminal tumors, again, if you look at each one is going to be compared to the standard of care, which would be neoadjuvant chemotherapy. Again, a product of the GU committee here. So the comparison for luminal papillary tumors, we're still really working on that. That's one of the to-do items, is to solidify what that would be like. But for example, right now we're thinking an FGF receptor inhibitor plus atezolizumab. And again, that'd be compared to neoadjuvant chemotherapy. For the basal tumors, we see a lot of response to checkpoint alone, so the atezolizumab, and then adding an anti-TIGIT antibody, comparing the neoadjuvant chemo. And then final, in the non-luminal, non-basal group, we cited the best comparison would be to atezolizumab and chemotherapy versus chemo alone. The primary outcome would be pathologic response. And you can imagine with this kind of integral-based biomarker study, the first bit of it will be an evaluation to make sure we have good feasibility, that we can reproducibly biomarker evaluate a patient, and then put them into a group based on their subtype. And then once that's going, again, a futility analysis at 50% of patients assessed to see if we're getting response. Now, we sent a survey out to the GU committee, and we appreciate everyone's response to that. 90% of people who responded said that they would be in favor of this kind of approach. So we're excited to be able to move forward with it. Next slide. Just wanted to give a little bit on some of the rationale of why these therapies for each subtype. Next slide. So this is, for example, the basal tumors, and this is early data on muscle invasive bladder cancers. This is actually from the PURE-1 cohort where patients were treated with pembrolizumab prior to cystectomy. And you can see from the red and blue bar graphs, if you just focus on that first group, the basal squamous tumors, they had a 70% pathologic response to pembrolizumab in this setting. If you compare that below to what was reported for Coxin, that was a 45% response. So that seems to be significantly better. Additionally, if you break that down further and you look at the TIGIT expression marker, I have that highlighted on the whisker plot next to it. You can see in the green highlighted group, those are the basal squamous tumors. TIGIT's very highly expressed in the basal tumor. So we're hoping that by putting those together, again, we'll see a synergistic response. And again, in our survey of patients, of providers from SWOG, we know that 65% are supportive of at least an anti-PD-1 for the basal tumors. Next slide. So for the luminal tumors, it's no surprise that we all think that FGF receptor 3 is going to play a major role. So this is expression, again, from PURE-1 of FGF receptor 2 and FGF receptor 3 expression. I highlighted in orange and red the expression level of both of these, and you can see it's very high. There's also a precedent in the metastatic setting. So FORT2 was described at ASCO and NORSE2 as at ESMO. And again, they're seeing when you add an FGF receptor inhibitor plus a checkpoint, you're getting objective responses anywhere from 50 to 70%. So we think this is going to be a good combination to put together for the luminal papillary tumors. Next slide. Finally, for why combine chemo and IO? Well, there is a benefit. And so this was just presented most recently at the AUA. This is Joachim's abstract on the Javelin 100 sort of genomic evaluation. And you can see the one group that really responded the best are the luminal infiltrated tumors. And we think those will probably be in our non-luminal non-basals, and they had a significantly improved response to serial therapy, but we think, again, by putting them together, this group may actually have a benefit. Next slide. So where do we stand with the classifier? So we're still working to solidify that. There's 3 vendors that we're currently working with, and there's a lot of things to be compared. You know, there's cost, there's speed, and there's the platform, and then what you can do with that. So, you know, in addition, many of these groups, while Decipher already has a FDA-approved biomarker, we we still are not all these groups are ready for that. So we're still working to kind of solidify that. We asked, again, our providers what they would want to see in response. 57% said they want to have a biomarker answer within 2 weeks, 40% by 4 weeks. So we think that, you know, that 2 to 4 week range is going to be really important for these platform results to come back in order to subtype a patient to get them on trial. Next slide. So we're also looking at what sites. So as Dr. Albing described, this is within SWOG. And so we know that we have good accruers for muscle invasive bladder cancer trials. So 10, 11, and 13, 14 were a very strong precedent. And so we know many of those sites are going to play an important role. But there's also the opportunity to reach out to other non-SWOG sites that have played a role in that trial. Additionally, there's a possibility to reach out to other trial networks to include them. At the bottom, you see the response on the survey. And again, this was providers suggesting how many patients per year they could potentially put on a trial like this. I think part of this trial, the fact that we're trying to look at many different therapies, that we'd like these trials to accrue very quickly to address these things, because again, the field is moving very fast. So accrual is going to be a critical thing. Next slide. So what do we think is going to be coming up in the next 6 months? Obviously solidifying the vendor. I think that the last target to identify is the best therapy for the luminal papillary tumors. Obviously the protocol needs further development, as well as some biomarker analysis, and of course, review by the executive committee. Thank you again.
Great, thank you, Josh. Rick had a question, and yes, we'd love to get you involved in this. I hope that's okay. Rick Bangs, and then Ian, you had a question. I don't know I think Kathy's probably still on. Kathy Albane, Ian.
Yeah. So just a philosophical question to bring up. Kathy and Lucky, maybe you can answer this for us. We have an exceptionally well-developed queue of studies that are on hold, waiting. They've gone through a high bar merit review. We oftentimes get 8 to 15 applications for our RFA process, and those studies are in a queue waiting to be developed and to be launched. It strikes me that we use some of the same resources for the CTP trials as we use for our GU committee trials. And just putting myself as an investigator in the GU committee, the opportunity to go through CTP versus going through our RFA process, how are we going to reconcile that merit review? And are there ways to get additional resources so the CTP doesn't use the resources of the GU committee and then, if you will, encumber somewhat our ability to develop the merit-based reviews that we go through currently? I'm just interested, Lucky, you know the process that we use. Maybe you may want to comment as well.
Sure, I'll have Kathy respond first about the process by which CTP.
But it's really, it's not a process by which CTP, because if CTP approves it, how are we going to apply, for example, our stat center resources?
Oh.
Kathy can answer that.
Yeah,
Because there's a
firewall. There's a
Firewall.
firewall. Is that what you were going to say, Lucky?
Yes, I was going to say
Yeah,
there's a
same thing. Go ahead, or I can. You go ahead.
So Ian, I don't want anyone to conflate NCI, NCTN trials or concepts with CTP concepts. There's a big fat firewall separating those two. Otherwise, we'll all be wearing jumpsuits in orange, right? CTP and PPP was designed by Kathy and the team specifically to ensure that there's no conflation of those two elements. And so the prioritization of concepts going through NCTN, NCI funded trials will remain as they have been for the past 50 years.
No,
CTP is a new mechanism.
I understand, Lucky. The question really is a matter of, we have Kathy and we have Eddie and we have Melissa. They're the ones who are, as well as their staff that help develop the trials. And we have this long stack of incredibly important concepts. In fact, there are investigators that are on the call today that and maybe this is not the place, but just something to think about.
We are
Incredible
thinking.
number of investigators with great plans, that we can only accomplish a certain number of trials, and the CTP trials use our same statistical resources to get them launched.
Ian, we have increased FTEs that are being hired under CTP funding. They are not going to, I can't, it's probably should talk offline more with you about it, but from Mike LeBlanc's perspective, Dana Sparks' perspective, the group chair's perspective, all of your issues are being considered and addressed so that it does not derail anything you're doing scientifically on the NCTN
So this
side.
won't have any impact on our current stat team?
No, or on your slots or on it. And Lucky, it's
Correct.
totally.
It's not a
10%.
slot issue, Kathy. It's a
What's the
bandwidth.
personnel that supports your queue, I think is what you're saying.
So there will be no impact. Okay, sounds
Well,
great.
Well, right now, because the ops office is down everywhere, we're trying to address that, and new hires are coming on board, not just for CTP, but on the NCTN side.
Thank you. Yeah, thanks. This is obviously a really
I don't want to.
topic, but you might.
Let's go on. I'm just.
May I move on? Yeah.
Yeah.
So just to, we're down to about probably 10, 15 minutes. So we're going to have to move pretty quickly to, we're not going to be able to cover everything. I've had this slide up. I hope everybody's aware. This is an open FDA meeting. It's a workshop, November 18th through 19th. This is the topic. I've got the link in here. Feel free to take a snapshot of that so you can sign on. It's relevant to the next topic. that Scott Delacroix, are you here?
Yeah, I'm here. 2 minutes or less.
By the way.
This is, can you hear me, Seth?
Yep. Yeah.
This is just representing our concept for a phase 2 trial for combination therapy in BCG unresponsive non-muscle invasive bladder cancer. A lot of people, multiple investigators are working on this. Next slide. As you know, the prospectively evaluated CR rates in BCG unresponsive space are modest, I think would be a reasonable descriptor. Even though we have made advances over the course of the last decade, they're still modest and hopefully room for improvement. Next slide. BCG still is going to be the first line of therapy. And instead of doing another single arm phase 2 trial, our thought process is going down a phase 2 multi-arm combination trial where we pick the winner, and the winner from this phase 2 trial, multi-arm trial, will then be chosen to advance to a phase 3 comparator trial. Next slide. Well, this didn't come through too nicely, but don't worry about the actual individual boxes, because we're working with industry, exploring some who's going to be in which box, but up to 4 different subgroups, all assessed. At least currently, the FDA mandate would be a biopsy for CR at 6 months, and assessing based on efficacy on that endpoint. Obviously, looking at safety with some of these new combinations, at least with the first 10 or so patients that go into each of these combination arms. Next slide. Next slide. That didn't come through too nice. Keep going. So pick the winner is going to really be based on CR unless there's any new guidance from the FDA. And if there's one is not better than the other, then we look at combinations that perhaps have less toxicity or lastly, less cost. Next slide. And this would be the phase 2 then going into below the line, which didn't project too nicely, a phase 3 where the winner from the phase 2 is chosen to be in a phase 3 arm against the standard of care. Next slide. All right, that's it. 2 minutes or less.
Great, thank you very much. I see you've got the advertisement for the trial, the program on this slide here. So this is a work in progress. I want to thank Peter Black, Parminder Singh, and a number of other people on the Bladder Committee who've helped contribute to this, and we'll keep working on this in the background. But I like the trial design, and it's we'll see what happens in the workshop, what comes out of that henceforth. So we'll press ahead. So Guru, is Roger on, or do you want to present this real quick?
No, I can. Yeah. So thanks, Seth. So real quickly, this is a concept for combination Ramishiromab and Nivolumab as adjuvant therapy for high-risk muscle invasive urothelial cancer. Next slide. I don't want to spend too much time, but I think we have a strong rationale for this combination, which looks active in metastatic disease, of course. And next slide. And we know that VEGF inhibitor plus chemotherapy seem to improve some outcomes, at least, as we know with the trials that did Gemcis bevacizumab. And Dan, you were involved in the Ramishiromab docetaxel, where there was a signal, but I know it did not achieve survival. Next slide. So Ramishiromab plus PD-1 inhibition is feasible. This has been shown in other settings. Next slide. And there are trials ongoing now. One of them that we will discuss here, I guess, with switch maintenance first-line metastatic disease, where cabozantinib will be looked at. And there is an ongoing trial of lenvatinib pembrolizumab in the first-line setting of metastatic cisplatin ineligible disease. Next slide. So next slide. I think I don't need to repeat the Checkmate 274 data, which showed adjuvant nivolumab improved outcomes. And so this design is as follows, quite simply: adjuvant nivolumab placebo versus nivolumab plus ramucirumab with an endpoint of DFS in all comers. And so at the moment, the way the study is it's a phase 3 design as it's written now. I'm in communication with Eli Lilly, they are thinking about it, but it looks like there's some inkling of interest, but we won't know anything till sometime early next year. So I think the RFA is also not out, so we will pursue that hopefully next year.
Great. We're not going to do this. Any questions for Guru real quick?
I think this will be a good design for a CTP also, because this space will lose the space if we don't act on this, and industry would jump on this if we can find a good partner.
Great. So just a reminder to join us on the Bladder Committee monthly calls. We'll be taking these two last concepts into additional discussion and review, and really would like to get your input. So I'd like to turn this over to Parminder for an update on 1806.
Sure. Thank you so much. Next slide, please. So quick, we all know the study design. Next slide. Just want to update. Thanks all for the support for this trial. I want to bring it to the attention of the new trials which are opening up. Congrats to Guru and Sarmad. And when we started to design this trial, I did a survey, and I got the number 10 of an average which I anticipated to enroll in a month. And so I took the range 8 to 12 in my study design, and now we are close to 7.5 after 2 years on an average of a month accrual on this. So I think those surveys serve good purpose, gives you a good idea of the accrual on your trial. So anybody who's planning a new trial should look into commitment from the sites, so then you can get an idea of how quickly you can accrue on your trial. So and I appreciate the support from Scott, Abhishek, Amit, and Josh for their great contribution to the accrual here. Thank you so much. Next slide. The key update which I want to talk about today, just for 1 minute, is that we brought this to the group's attention in the beginning of the year that we are accruing more T2. The trial was designed with T2, T3, T4 as 50-50 stratification. The intent was that we wanted to get more events if we have a higher T3, T4 numbers. We were close to 10 or 11% T3, T4 and 88 to 90% of T2 earlier in the year. Me, Seth, and Jason, we did a chart review and we pulled that number to 80-20. But we certainly encourage sites to enroll T3, T4. We had initially thought that we might get more advanced disease patients onto this trial, but we are seeing that more T2 patients are going on to this. And so please look at the original op notes for accurate clinical staging, because many times the initial TURBT, which showed an 8 centimeter mass or a 9 centimeter mass, was done outside, and by the time the patient comes to you in the hospital, there is no tumor in the bladder. And so I would encourage to look at the original op note for proper clinical staging. Look for prostate involvement in the biopsy. I just had a patient who had prostate involvement in the biopsy report and was staged as T2. So we requested that patient to be re-stratified. And so also encourage your urology partners to do an exam under anesthesia to get an accurate clinical stage. So that we can encourage more T3, T4 patients to be enrolled onto the study. The other key update is that, you all know that I have been struggling getting the true culture specimen collection moving on this trial. We had initially selected some sites which we thought will get more samples, but now what had happened is the vendor which provides the tube, they have changed the solution in the tube, and we haven't done the bridging study. So Genentech has asked us to halt the true culture specimen collection altogether. And so we will send that as a memo to the sites who were on for collection, especially Scott. We are going to hold further true culture specimen collection. The third important key update, which people who are on the bladder call, they know that Seth had been working closely to get a ctDNA idea built into the platform because of the data coming out of IMvigor. So we are going to add, next slide, Josh is helping me add, if you see the box on the right top, add 2 more blood sample collections so that we could do ctDNA and also urine free DNA in these patients. So it might come to you as an amendment moving forward. And also, since we are going out of the true culture, David is interesting in bringing to the table B cell or T cell receptor types to be added to the TM. So that's something to be coming in future. Thank you.
Yeah, thanks, Parminder. This trial is accruing at a rate that I think we can all be very proud of, and kudos to the team for moving this along. A very quick update from, I don't know, is Peter on?
I'm here.
Yeah. Anything that you want to communicate to the group?
If you just go to the last slide, so the 1605 is 1 year of atezolizumab. Sorry, that one, yeah. For BCG unresponsive disease, we presented 18-month data at ASCO this year, and you can see on the left the duration of response after 6-month CR in the patients with CIS, and on the right you see the event-free survival in 55 patients with no CIS, so papillary disease only. So a median response duration in both cases of about 15, and some of the event-free survival rates are listed in the table at the top. So the bottom line is that the results are very similar to other drugs in the same space, and so we are preparing this for publication, and Genentech, of course, is looking at it. So I'll finish there.
Great. Yeah, exciting stuff. Thank you very much, Peter and the team. Rob, are you there?
Yeah, I'm here. Can you hear me? Just an update on S1602. We completed, can we go forward? So I also have to give a shout out to Scott, who really helped us in that early period when we were just getting moving. If not for his group, we that first year would have had hardly any patients. We had a couple of bumps. You can see that the accrual dropped on 2 separate occasions, but we ended running to the tape. And I want to thank all the investigators. Could you go to the next slide? All the investigators, particularly those listed here, really did the yeoman's work here in putting these patients on trial. We overshot our target. We ended up with 1,000 patients. We are now, can we go to the next slide? We're now in the process of reviewing data, hoping to meet with the FDA in early part of next year to consider a pathway forward for approval of the product based on the CIS response rate, which we're hoping will be comparable to TICE and give us a pathway forward. So this is mainly just thanks to everyone for their hard work in making this trial happen. Thanks, Seth.
Thank you, Rob. So in deference to the rest of the team, I'm just going to cruise through this. This will be very speed-like. If any of the investigator PIs need to say something, please do so, because we need to get on with the prostate session. So no updates on 1011. I talked about the TM yesterday. Tom, Flag, we have the PFS and OS data. There was an abstract submitted to GU ASCO, and there'll be a manuscript in the works. You heard all the exciting TM stuff that's in the works with David McConkey and the number of investigators on this trial. Just a reminder, Ambassador closed early, although it had come within about 30 to 40 patients of completing accrual with the approval of Nivolumab, which would be a standard of care in this space. So one would expect that there'll be some really strong data that comes out of this. Andrea Paula, who's the PI, indicated that they're looking at the analysis now, but stay tuned on that. Guru, thank you for being our SWOG champion on that. Please continue to support this alliance trial. This is patients get GC. This Gopa Ayers, the PI, Amit Gupta, Shilpa Gupta are our SWOG champions. This is using a panel of DNA damage response gene alterations. If there's one or more alterations and a complete response or only residual CIS, patients can be observed, which is a very novel approach. And that would occur in about 25% of the patients. This still has a ways to go. 121 patients accrued out of 271. So please continue to support this. This is another trial in the BCG unresponsive space, an alliance trial. It's intravesical gemcitabine plus Pembro, thanks to Chetham Ramamurthy from San Antonio and Mark Tyson from Mayo Scottsdale for being our champions there. Again, I think Scott Delacroix is killing it on this trial. We've just got this, we're very close to getting it activated and expect to be able to support it. And then, as you heard before, we're really working hard to try to get ourselves back into this space on the heels of 1605. Josh, any words about Monica Joshi's trial, bladder-sparing chemoradiation therapy?
Yeah, really need to open this trial. I mean, this is a trial addressing a really important need, trying to determine multiple features as far as systemic response, radiation response for people locally advanced medicine, local nodes. And so please consider opening this trial. It really needs our support from SWOG. I believe there's 2 patients currently enrolled, so it needs some support.
And this should not really compete with 1806. And then Shilpa, a quick word here on your.
Yeah, thank you, Seth. So this is the main CAV study that is looking at maintenance avelumab versus cabozantinib and avelumab after frontline platinum-based chemotherapy in metastatic urothelial cancer patients who do not progress. It's 654 patient trial. Primary endpoint is overall survival. And we have a variety of correlatives, including DNA, RNA-seq, immune biomarkers, ctDNA, urinary stool samples. So Ed is the SWOG champion for this. It got CTEP approved just last week, and it's under CIRB review right now. And the OEWG date is March 22nd of next year. We are on track with the timeline. Thank you, sir.
Shilpa had the wisdom several years ago to put this forward, and you really stuck with it. So congratulations on getting this approved and then pivoting at the last minute when Nivo got approved. So really hats off to you and the team at Alliance, and thank you for bringing this to us at SWOG. I think Dan, you asked for 2 minutes. You can have 1 if you don't mind. Are you on? Daniel Vena, are you there? Maybe in the audience, but he did want to mention this Alliance trial, which is duration of immune checkpoint therapy in locally advanced or metastatic disease, a randomized phase 3 non-inferiority trial. And you see the study chairs down at the right. Dan's our study champion, and please reach out to Dan if you have any questions about opening this trial. I think that is it. Thank you very much for the extra couple of minutes. Really appreciate all the contributions from our great bladder committees, stats and ops, and thank you all very much. Appreciate it.
Great job, Seth. Thanks so much. It's an impressive group. Dan and David?
I'm going to try and share.
When he's sharing, yeah, thanks to everybody, especially the prostate group that meets monthly on first Friday of the month at 8 a.m. Pacific, 10 Central. So welcome anyone who wants to join that while David's getting the slides up. Similar to the bladder group, we're going to run through mostly the active trials and some of the metrics, and also present some of the data from some of our past ones. We have updates at the end on all of our intergroup trials. We likely won't get there if we're aiming to end in 40 minutes or so, or an hour, and but we'll do our best. So Dave?
All right. Can people see my screen?
Yes.
Okay, that's a relief. Okay, good. We're going to move forward post haste. This is the agenda, which may end somewhere in the middle here. And I would like to try and review the first three other group studies we're doing. We'll see whether we get there. I'm going to go straight across to Brian Chapin. Brian, take it away on 1802.
Yeah, so thanks for the opportunity to give everyone an update on where we're at with 1802, which is our randomized phase 3 trial of standard systemic therapy versus standard systemic therapy plus definitive treatment of the primary. Next slide. So just a reminder of the trial schema for those of you who are not familiar. This is castration-sensitive men undergoing systemic therapy for up to 22 to 28 weeks, standard therapy by NCCN guidelines, and then randomized to undergo continued systemic therapy with or without definitive treatment, and that can be surgery or radiation, and then followed for an overall survival endpoint. Next slide. David?
I'm pressing.
Oh. Maybe push the, yeah. So this is the accrual as of October 1st, 2021. We have 440 patients. We're about a third of the way there. As you can see, the numbers have kind of gone up and down in response to various things, including COVID and the lack of staff, lack of ability to open up at sites. It seems like when we do get on our sites up and rolling and COVID's kind of in its low, we do fairly well. But we have those periods where we drop below that dotted accrual line that we'd like to be making. You can see here at the top a list of the sites that have been very active. And if we go to the next slide, those are a little bit more visible here. And again, we just were commenting in the chat box about Scott Delacroix and Jesse Gills and the crew at LSU and how well they're doing with accrual to all studies. And they're leading this accrual at 56. You can see there's about 20 or so programs that have put on 3 or more patients. And I think that ideally we'd like to get those sites to add and continue to add to this protocol, but we'd really like to focus on the sites that have opened this and gone through the effort to get this trial up and going, but maybe have been slow to accrual. So any efforts that you need from our side to try to help with that, please feel free to reach out to me directly or to Dan or any of the other folks in the SWOG group to address those and potentially even have discussions if that's helpful with your sites. Next slide. So there's been questions people have been asking specific to participant characteristics, particularly related to how many patients are having radiation versus surgery. As you can see here, it's about a 60-40 split for those receiving radiotherapy. And then the other question that comes up frequently is, with Stampede RMH and with some reluctance on the radiation oncology side to put patients on trial who have low volume metastatic disease, what percentage of patients are actually low volume? And so about 50-50 split if you look at the polymetastatic versus oligometastatic definitions down in the bottom of this. And then if you go by charted, it's about a 60-40 split. So we are seeing low volume patients going on study, and we are currently seeing a lot of patients who are receiving radiation therapy as their local treatment. So for going forward, it would be helpful if the urology groups here could put patients on, particularly if you're considering a local therapy with surgery in the setting of metastatic disease. Next slide. So some of you might know and others may not, but we have a CME video that we put together for this trial with the support of the Hope Foundation. And this is a video that is about 45 minutes of a lecture on the topic of local therapy in metastatic prostate cancer, followed by a discussion between me, Dr. Eggener, Tony Crispino, and Dr. Delacroix about the difficulties and the things that we're looking at in this study. That will become available to people to use within their groups if they choose to, or even just through things like the SUO, possibly the AUA. We do have the patient education video, which I think people are still surprised when I explain it to them what that is, but that's available. It's basically a layman's description of what's going on with this study. We give it to our patients to review prior to their consultations in my clinic. And it's been very helpful from a patient's perspective to understand what the trial is actually saying. People don't understand randomization until they start to see that in front of them on a video. And then most recently, we have some protocol-specific materials that are coming through CTSU, which are a fact sheet for reference from clinics and a protocol card, and these will be available shortly, and you can have them available in your clinics to remind you of this study or your faculty of this study. And then we had a nice discussion yesterday at the executive meeting about some other additional opportunities for how we can improve enrollment, and we'll be reaching out directly to the NCORE sites and then trying to identify regional champions for this study so that we can get some more support from individual groups. And then, as I mentioned, all of the CME and the presentation, I'm happy to speak to any group about this, and we'll identify some additional people that can have access to any of my PowerPoints or anything they need in order to get a better understanding of the protocol and try to get things continue to move forward. So that's all I have. My email, my contact, and the SWOG study contact if there's any additional questions. And thanks very much for the time.
Thanks, Brian. Can you put the website in the chat for people for the video?
Sure can. And from a, just an ease of logistics, ease of use, it's on YouTube. All they have to do is search Chapin SWOG 1802, and it'll come up for like your staff at the sites. But I'll share the chat for sure.
Actually, I've searched, Brian. You have a few other videos there, but we can talk about that later. Okay. All right. Okay. Thank you. Now, Niraj.
Yeah.
With us, Niraj?
Yes, I'm here.
Okay, the question is, Niraj, whether I can press the button faster than you can talk. So give us
all
an
right.
update on.
Sounds good. So these are the results of SWOG 1216, which we presented in ASCO this year. This is a study design randomizing more than 1,000 patients to Lupron plus TAC-700 versus Lupron with bicalutamide. Next, overall survival was the primary endpoint. So the trial was designed, trial had 90% power to achieve the overall survival analysis to be significant. Next slide. Interestingly, the demographics showed that we recruited highest number of African-American men, 10% of all in any of the MCSPC trial. Median age was reflective of this disease population. Next. We can skip this. Next. So minimal versus extensive disease, evenly balanced, 50% and 50%. Primary analysis. We saw the overall survival in the experimental arm was 11 months higher than the control arm, very similar to what we have seen with recent trials. However, we missed the mark. P-value was 0.04, but confidence interval exceeded 1. After a median follow-up of 4.9 years. Next. Too many arrows here. Most of the subgroups benefited, except patients who received ADT prior to registration. I do not know why that happened, but overall, we can see the benefit across the group. Next. Progression-free survival is improved by twice as much, which is consistent with or recently reported trials in this context. Next. Secondary endpoints such as PSA responses favor TAC-700. Next. Adverse events are not surprising. We see hot flashes, fatigue, which are consistent with deeper androgen blockade. Hypertension was more common, again consistent with the mechanism of action of TAC-700. Next. Post-Hoc analysis, we decided to do that given insignificant results, despite having such a strong survival benefit, especially in the control arm. I will show you in the next slide. 80% patients, almost 80% patients received subsequent life-prolonging therapy. And this is only first line. I think I have a feeling that because most of the patients were recruited in these large American academic institutions, people could have received 3 to 4 lines of therapies on the control arm. Next. Next. So this is the comparison of the control arm survival of the across the trials. And we can see the control arm is lived for 70 years, the median overall survival, which is remarkable, great news for our patients. If you compare with the volume of the disease, of course, the definition of volume in the STAMPEDE trial is different from the definition of volume in the SWOG trial, but still, if you look at the volume status, STAMPEDE, abiraterone, docetaxel trials have similar number of patients with high risk or high volume disease. Next. If you compare with the SWOG 9346 trial, which was reported 10 years ago, and the proportion of patients at high risk by SWOG definition was exactly similar, almost identical, we see a 24 months improvement in overall survival in the control arm. And I think that's where the trial got hit for good reasons, I suppose. Next slide. So to conclude, even though we did not see any survival benefit, which was significant with TAC 700, I'll come to the last point. We still showed that I think this trial established a new landmark for survival estimates when patients with MCSPC have access to multiple approved subsequent life prolonging therapies. So, of course, great job by all the centers across the country. It would not have been possible to recruit 1,300 patients, despite having such a stiff competition from the recent approvals. So thank you to all of you. And we will publish the results very soon. Thank you very much.
Thank you, Niraj, and the team that worked on this. Excellent work. And I'm going to move on now as we run across the bridge to trials that are in development. Now, is Heather Cheng with us?
Yes, I'm here. Thanks so much. I'll try to keep this really brief. The first thing I want to say is I missed the time yesterday, so I wanted to just thank Dr. Vogelwein because I didn't have a chance to do it in the other session. But this trial is actually supported very much early on by his enthusiasm, so I wanted to especially acknowledge him. So this is targeted neoadjuvant treatment for patients with localized prostate cancer and germline DNA repair deficiency. If you go to the next slide, the hypothesis is that 12 weeks of neoadjuvant therapy followed by radical prostatectomy for men with localized unfavorable intermediate or higher risk prostate cancer and germline BRCA1 and 2 mutations will result in a higher pathologic complete response if treatment is molecularly guided with ADT and carboplatin versus not specifically guided with ADT as the control. And this will allow us to facilitate deeper translational understanding. I won't go through that. We can go to the next slide. So we can skip over this. This is really to just highlight that the NCCN guidelines now do incorporate offering men with high risk localized and higher germline genetic testing and genetic counseling. So this is the scheme of the trial. I think we've seen this before. It has evolved with a lot of extremely valuable input from everyone. I want to especially thank the urologists and our patient advocates and Kathy Tengen and the stats team. So unfavorable intermediate or higher risk disease, germline DNA repair alterations. We sort of focus primarily initially on BRCA1 and 2 and 1, but also are including CHEK2, PALB2, RAD51C and D. Local testing prior to enrollment is allowed. That's a question that comes up frequently. And they'll be stratified by NCCN risk, unfavorable, intermediate versus high risk in a 2-to-1 design with ADT as a control arm and 12 weeks of ADT with carboplatin AUC5 given every 3 weeks times 4 cycles. There will be translational biospecimens collected prior to systemic therapy, prior to prostatectomy and post-prostatectomy, as well as collection of our prostatectomy tissue. The primary endpoint is pathologic complete response, 5% null hypothesis. And we are acknowledging that there may be some, we don't know what the response rate will be for DNA, germline DNA repair carriers. There's some suggestions in the literature, but we don't have a lot of data. So we've built in with Kathy's sort of input a 15% difference between the two arms, given there is some uncertainty. So secondary endpoints are there and translational correlatives. We talked about yesterday in the TM session. Happy for your input. This is going to triage on Monday. So thanks so much, everyone.
Thank you. Thank you. Okay, we'll hear more about that. Do we have Paul Korn with us?
Yes, thank you very much for the time. Again, in terms of maintaining time efficiency here, this concept is well known to the group. Looking at these aggressive variant prostate cancers, as a quick reminder, it's the identification of these very aggressive tumors that have clinical features like small cell, but often the pathologic features of adenocarcinoma. So through our work and others, we've identified a tentative molecular signature for these bad actors that looks at 2 of 3 suppressor gene loss of P53RB and P10. And if you go to the next slide, Colin, we published a Lancet randomized phase 2 where when we did a post-hoc analysis, we appreciated that the benefit of platinum-based chemotherapy was particularly marked in the patients who harbored the aggressive variant molecular signature. So we put forth a concept for testing in SWOG on the next slide, where we wanted to do a larger phase 3 to further confirm the signature. And we're doing this in the castrate-resistant space, where we will look at radiographic progression-free survival as a primary endpoint, and we'll be measuring the signature at baseline and stratifying for that signature. The update I have for you, and I'm pleased to share, is that Kathy's was able to start helping us with the statistics. So the big picture is that it still remains about a 500 patient trial or so, but rather than the MD Anderson group, which had looked at primarily one interim analysis for futility, Kathy has very elegantly suggested some multiple interim tests for futility and superiority, and most importantly, at bullet point 2, use a step-down approach where we look first at the molecular signature positive group, where we are most confident about the treatment effect before deciding whether to look at the other groups. It's a term called using the same alpha without splitting it. It's a fixed sequence approach. So I know we'll have a chance to go into this at more detail in the monthly call. But David, I think what we're building towards is being near close now to perhaps going forward to the prostate task force again, with kind of the blessing of the group is what we hope to do next.
Fantastic. Thank you very much, Paul. And we'll look forward to trying to get this moving forward. And thank you to Kathy, who's been working as usual.
Yeah, very much. Yeah,
very.
It was absolutely fantastic to have her on board.
All right, so we're going to move along, and we're going to get a Mike Swizer, who I hope has also been able to get in, and he's going to give us a review of the Battering MCRPC trial proposal.
Yeah, thanks. I think you all can hear me. So this is a concept that we've been developing for a bit now. Can you advance the slide for me? So bipolar androgen therapy, or BAT, is a cyclic form of high-dose testosterone, results in T levels spiking up to supraphysiologic ranges and then declining to near castrate over a 28-day treatment cycle. This has been shown to result in clinical responses in about 1/3 of men with castrate-resistant prostate cancer, and these effects may be mediated by double-stranded DNA damage. In addition, preclinical as well as clinical data support that BAT may actually be able to suppress a number of key resistance mechanisms that drives cross-resistance between androgen receptor-targeted drugs like abiraterone or enzalutamide. Next. Next slide. There you go. Oops. So can you go back one? Sorry. This is the schematic for a phase 2 study, the Transformer trial, which I think a lot of the folks on the call were involved with, essentially looking at a low-risk castrate-resistant patient population who had minimal to no cancer symptoms and had previously progressed on abiraterone, randomized between BAT and enzalutamide. And the study also allowed for crossover with some secondary objectives aimed at understanding if BAT could in fact mitigate cross-resistance between Abi and Enza. Next slide. Overall, they showed that the outcomes with BAT or enzalutamide were remarkably similar in patients when they initially randomized, with PSA response rates around 25 to 30% and progression-free survival of about 5.5 months, regardless of which arm patients went to. Next slide. However, the thing that was most interesting was that there was clinical evidence that BAT could attenuate cross-resistance between AbbVie and Enza. In the figure on the left, the red bars represent patients that got enzalutamide after BAT, and they showed that in those patients, they had PSA response rates of 72%, and the progression-free survival for those that had the sequence of BAT to enzalutamide was quite long at 28 months. Next slide. And I think most importantly, when they looked at the outcomes in terms of survival for patients who had progressed on abiraterone and then randomized to BAT enzalutamide versus enzalutamide, they showed overall survival benefit of 37 months versus 29 months when you got BAT enza. And just to put that into context, that correlates with over 50% relative reduction in the risk of death, which compared to both AbbVie and Enza when they were tested in the pre-chemotherapy space only showed around a 20 to 30% relative reduction in death. So it looks quite favorable in that respect. Next slide. And so that formed the basis for this concept, the battering MCRPC study. We're looking at a very similar patient population to Transformer. So patients with lower risk castrate-resistant prostate cancer, they have to have minimal to no cancer-related symptoms, previously progressed on abiraterone, and then they randomized between BAT followed by enzalutamide at the time of progression versus enzalutamide alone. And the primary endpoint is overall survival. So next slide. So that's sort of the study in a nutshell, and I'd welcome any feedback if anybody perceives any challenges in operationalizing a trial like this.
So this is very interesting, innovative, and follows on some interesting work, and we're trying to get the biology sorted out. Just at the moment, I don't think we're going to be able to entertain questions for reasons of time and what have you, but we're going to put a survey out because just to work out what people's feelings are on this, how they feel about given the BAT and running the trial, and then we'll look at this. We'll look at this for potentially the next RFA and look forward to that. So in the interest of time, I'm going to move on, and I do take Niraj Agarwal's comment that the patients will like the trial. So we just need to get them through the randomization stuff, and we're going to be asking you all about that. Okay, nice job. So we're going to change pace a little bit, and we're going to go to Austin Kirshner, who's presented to us before. He's from Vanderbilt. He's a radiation oncologist, and James, you brought this concept back to us from the newly formed radiation committee. So Austin, do you want to take us through your plan for this randomized study of immunoradiation therapy and oligoprogressive osseous metastases, the IO-CARE study? And are you with us, Austin? You got
Yes, I'm here. Thank you.
Good.
So I appreciate the opportunity to talk about this concept. As we all know, immune checkpoint inhibitors are not widely used for metastatic prostate cancer, but there's a lot of interest in this. A lot of combination therapies are being looked at on multiple clinical trials, some of which have some reasonable signal, but some of them are failing to show data. But I think that as a radiation oncologist, we see plenty of these patients who need treatment for bone metastases. And there's a population which may have an oligoprogressive state that we could capitalize on to try to help ablate disease and cause an immune stimulation. So if you can go to the next slide, there was a study that was done on this a few years ago out of Bristol Myers Squibb sponsorship, where they did 8 gray of radiation to up to 5 bone lesions in patients who were progressing after docetaxel for metastatic prostate, a castrate-resistant disease. They gave 8 gray to both arms, but one arm also received Nivolumab as the checkpoint inhibitor to see if it would stimulate immune responses. They could maintain on maintenance Ipilimumab as well if they don't progress. So there, if you go back 1 second, there was a tail there that was just an update showing that there's some persistent responses in immunotherapy for the long term. So this has some potential durability of response. So on the next slide, what we did was we looked at a sub-analysis, the trial authors looked at a sub-analysis that showed that favorable patients who have limited disease burden seem to have the greatest median survival increase over 7 months, which is significantly higher than a lot of the FDA-approved drugs for the metastatic castrate space. IPI alone, also on the top right here, doesn't show a signal. So it really is the combo of the 8-gray radiation plus the drug that seemed to give a signal. But the challenges that we wanted to address was that ipilimumab has significant toxicities, especially at the high dose they were using back then. And we thought changing to a PD-1 or PD-L1 agent would be superior in toxicity. And then increasing the responsiveness through a better way of doing the radiation would be an improvement as well. So on the next slide, what we did in my laboratory was some preclinical studies using cell cultures as well as mouse models for prostate cancer castrate-resistant space. We found that there's a peak stimulation of the STING pathway through the expression of cytosolic DNA between the 10 to 16 gray range. So single fraction doses in that area seem to provide the greatest stimulation of production of type 1 interferon. And if you can see in the middle figure, the trial was using 8 gray and 1 treatment on the left there. And we're proposing that we use 13 gray times 2 with a every other day approach to give us the more stimulating immune response. And on the right, these are clinical doses that are used on trials and in current clinical practice for radiation oncology. So it still is in the realm of doability of clinical practice to do this kind of dosing. On the next slide, what we did also is show that in mice, we had a increased survival by combination of PD-1 or PD-L1 agent with radiation given in this range, 10 gray times 2. There was an overall survival advantage. And on the next slide, we saw that it's actually mediated by a CD8. So if you block CD8 T cells, there is a loss of that survival advantage there on the right. And we're seeing that tumors that are irradiated in mice, as well as the distant tumors, when we treated one tumor of 2 tumors in a mouse, both tumors have increased CD8 T cells when they respond to the treatment. So it's a very interesting thing that we're demonstrating some immune responses with this higher dose of radiation. So on the next slide, this is our scheme of proposal. We would like to suggest that patients who are oligoprogressive, meaning they have up to 5 new bone lesions in the metastatic castrate-resistant space, could be enrolled on the trial. They could be receiving their standard systemic therapy on which they're progressing up to 5 lesions. And we treat all of them with radiation to ablate the lesion, that SBRT style, 13 gray times 2. And one of the arms has the addition of the checkpoint inhibitor. And we haven't yet selected exactly which agent it would be. It would depend on what we discuss with pharma and feedback from the group. Genentech had given us an initial positive feedback, so we had suggested atezolizumab as the agent, but I'm open to all suggestions. Radiographic progression-free survival is the endpoint, so we would have bone scans done before treatment and about 3 months after treatment to see what the status is of the treated lesions, as well as any distant lesions that might change. And they would be enrolled in a sort of dynamic minimization approach with 42 patients per arm to try to equalize the prior history of treatments, since these can be diverse patients who've had different levels of treatment for their castrate-resistant disease, whether it's a next-generation androgen deprivation therapy or chemo or other agents. So on the next slide, we just want to emphasize progression-free survival by radiographic means is going to be the primary endpoint. And we think we can achieve this just simply by bone scans. We're aware that there are much more modern approaches like PSMA scans, but the most widely available would be a bone scan that we'd use to quantify data. And this has been recommended by the Prostate Cancer Working Group trialists. So there's just an example on the bottom where you can take a scan before treatment, one at the end of treatment of the 12 weeks or so, and then use that scan as a new baseline. And if new lesions appear in the 2+2 paradigm, then you can call progression once you're sure that there's absolute progression of disease. So on the next slide, we would accrue for about 2 years to achieve 84 patients, estimating that progression-free survival, we'd like to see an increase at 6 months of about 20%. And on the right there is the data from the trial that I mentioned with ipilimumab, that they showed an increase of about 12% progression-free survival in these heavily pretreated patients with a 6-month PFS up to 30%. So we're hoping to at least meet that or beat that with this slightly improved radiation approach and switching to a PD-1 agent. And go to the next slide, please. So that's the summary. We would like to propose this as a potential opening for oligoprogressive disease. PD-1, we have preclinical data that shows an improvement, and we could randomize patients to all be receiving radiation for their needed bone treatment, and then some will get the checkpoint inhibitor. And we also want to propose biomarkers to go along with this study. So collecting circulating blood for cytokines, blood counts, circulating tumor cells, potentially T cell profiling or circulating tumor commercially done. And the next slide. we have, I think there's some questions about the design, but there are lots of folks who've been involved in this, and I appreciate them all. If you want to go to the next one, the alternative designs that we wanted to talk to the group about could be doing things at the end stage, like they did on that IPI trial after failing standard therapies like docetaxel and other agents with no other standard options. They could be enrolled. Number one is the design we currently think is reasonable. If patients are at any point in their castrate-resistant space just progressing in a few lesions, those can be sent for radiation. But there's also discussion about standardizing systemic therapies that perhaps patients who are newly entering the space of metastatic disease from being non-metastatic to metastatic could be eligible for treatment on this trial to try to maintain the current line of therapy that they're on. So if you have a patient on AbbVie who's non-metastatic, becomes oligometastatic, they could potentially ablate those lesions and then continue on the same drug. Or we could do a standardized drug at the bottom there, number 4, to just switch whatever agent they're on to some standard agent that we would say is the next line. But these may limit the eligibility of certain patients who've had prior treatments. So thank you very much. I'm open to all discussion and feedback about this.
Can I ask a quick question? Seth Lerner here. Really nice work. Congratulations. 2 questions. The simplicity of bone scan, I get it. But since I don't take care of these patients, is PET-CT used in this disease state? A little, a lot, because that would obviously have an impact on your trial.
I think it's not standardized that patients should get that type of imaging, but we can offer patients PSMA PET scans now that they've become pretty widely available. And we just got it at Vanderbilt a couple months ago. So it can be used. I just don't know if we should be doing it in the metastatic castrate space.
And could it be bone plus, say, soft tissue, or bone only?
I think it should be bone only, just because that population is the most common and the ones that seem to do the best on that IPI trial. The visceral disease patients did poorly, and lymph nodes are a little ambiguous. It's not visceral disease, but we usually don't need to radiate a lymph node for some reason. It's more of a bone issue for palliation.
All right. So I think the issue with the novel scan, Seth, is what progression means if you do them sequentially, and also paying for them. So we'll work on that. Now, Abhishek Salinki had a good point about trying to get SBRT approved for metastatic castration-resistant patients. And that would be an issue we have to look at. It's not available everywhere, but it's widely available. And generally, Medicare smiles upon us when we do an NCI designated trial. So we have a deficit of time, and in this format, problems with getting Q&A back and forward. So we're gonna follow Seth Lerner, and we're gonna send the survey out, and we're gonna put a special tag on it to make sure Seth responds on both of these prostate concepts. And we're going to look at what people think about this. We do have some stuff to work through here, but I think both of these concepts are interesting, and we also need to get the trial done. And the questions came up as to how we get this done. This is a small trial. I think it's an important proof of concept that's ideally done in SWOG, and we need to work out whether we're going to do this through the radiation group, and they're going to lead it. Or whether the new mechanism, with help from our very good friends at Genentech, may be a way of getting this done. So we'll hear more about this. In the interest of time, I'm going to move it along. And thank you very much, Austin, for a very interesting presentation. Okay. All right, now, Earl Burgess.
Thank you, David. I wanted to raise awareness to the group about a study that's been in development that should activate within the next 1 to 2 months. This is SWOG 2012. It's a randomized phase 2/3 trial of platinum etoposide with or without atezolizumab in patients with metastatic extrapulmonary small cell cancer. The study is being led by David Jen, who's a GI medical oncologist at the University of Washington, and co-chaired by Elena Chorian, with additional study leadership here. And so, based on the experience in small cell lung cancer, this study is seeking to ask the question of whether adding an immune checkpoint inhibitor, atezo, to a backbone of standard chemo also shows benefit in extrapulmonary small cell. Next slide. So here's the schema. The key eligibility criteria, as I've mentioned, are that patients have to have extrapulmonary small cell carcinoma. So for bladder and upper tract, upper urothelial patients, mixed small cell and urothelial cases are excluded because of the use of the standard use of immune checkpoint inhibitors in urothelial disease. But for patients with prostate cancer, in particular treatment emergent small cell carcinoma, a prior history of adenocarcinoma is allowed. However, for patients with suspected treatment emergent small cell prostate cancer, an additional biopsy proving small cell histology is required. Patients are allowed to have no prior treatment for their small cell component except for one cycle of bridging platinum etoposide. So again, for relevant to prostate, for patients who have treatment emergent small cell prostate cancer, concurrent therapy with ADT is allowed, as well as apalutamide and enzalutamide after the induction phase, but abiraterone and prednisone will not because of the prednisone component. Patients will be stratified by performance status and site of origin, with prostate being one of the specific stratification factors. So patients are randomized 1:1:1 to one of 3 arms, all of which are based on a standard platinum etoposide backbone. And the 2 experimental arms that I'll just point out is the addition of atezolizumab in the induction chemo phase for both, but one of the experimental arms includes maintenance atezo for up to a year, whereas the other does not. Next slide. And so just a few more comments about the study design. The primary endpoint is overall survival, which is in part hierarchically tested with the initial analysis based on the standard of care chemo alone versus the combination of atezo with induction followed by maintenance, with subsequent pre-specified testing as are shown here. And so the secondary endpoints are shown, and tissue will be banked for future biomarker analysis. The phase 2 analysis will occur after 50% of the phase 3 information is available, and it has built-in stopping rules for futility, and is expected to enroll 190 patients over 3.5 years. So that equates to 4 to 5 patients per month, which is in line with contemporary studies. So the null hypothesis is based on a 12-month landmark overall survival of 35% and is powered robustly to show an improvement to a hazard ratio of 0.53 in the atezo arm with maintenance. Next slide. So here's the current status of the study. It's been approved by the committees that you can see here. CTEP has already approved protocol. In fact, has been submitted to the central IRB. We received the initial feedback earlier this week. The comments were minor, and so resubmission to the IRB is expected within the next couple of weeks. And currently the study is projected to activate within the next couple of months. So just wanted to make everybody aware of this study, that it will be activating very soon. Would ask that you speak to the additional disease lead key stakeholders at your institution, and please consider opening the study so we can ask and answer the question of whether the addition of atezo to the chemo backbone is also beneficial in extrapulmonary small cell. So we've seen a similar experience in small cell lung cancer. Thank you.
Thank you, Earl. We all see these patients periodically. Obviously, MD Anderson have done the speciality in this over the years, and I hope that we can contribute well to this. So please be on the lookout. It may be opened at your institution by someone else, like a GI oncologist or a gyne-onc or somebody. So just be aware that this is around.
Could you repeat the eligibility again? What's not allowed as prior therapy and what is?
Sure, and then just a comment on the question as well about the histology. So pure small cell bladders are allowed. Mixed urothelial small cells are not for urothelial disease. In terms of prior therapy for small cell directed therapy as it relates to prostate cancer, can have standard of care for prostate, but no specific therapy for patients that have treatment emergent small cell prostate cancer, no prior therapy in that setting. So you can imagine if they've had standard ADT plus and then developed treatment emergent, as long as there has not been prior therapy for the small cell component, they are allowed.
So you said earlier they could not have received abi-pred because of the pred. How about docetaxel pred?
Sorry, that's the comment was specifically about concurrent therapy on studies. So based on the assumption that some people may wish to continue to treat the
got it.
carcinoma component. Yeah. So,
Right. All right. So does that address your question, Dr. Lara?
Does.
Okay, thank you. All right. So you'll be seeing this soon, probably in the next 2 months, we think. And we'll try and also forward some sort of link or something to let people know about it. And it is a good study to be done in SWOG. Okay, thank you very much, Earl. Now this got the award as the fat bear coming out of hibernation, I think, in Canada. And so we're at a point where I need to ask Ian Thompson, and I've got it at 1:20 here on the West Coast. Should we proceed a little more, or do you want to give me the yank?
Oh, you have another 10 minutes, and we do have a buffer, so.
We have a buffer. Actually, this bear has a buffer as well, but we won't get into that. All right, so ongoing studies, and we'll keep going until we get the yank. And Evan, the Indicate trial, EA 8191.
Thanks, David. So this is a trial from ECOG Akron, and it's a trial based on patients who have had previous radical prostatectomy and are undergoing salvage radiation therapy. So there's really two questions being asked here. One is there a role for enhanced systemic therapy, and they're using apalutamide here to add to the standard androgen deprivation. And the second is the question about metastasis-directed therapy. So currently it's using Adjuvant PET, but there is an amendment being written right now to allow PSMA PET. It won't change the PSA criteria, which is a PSA of 0.5 generally. But if you relapsed in less than 1 year from radical prostatectomy, a PSA of 0.2 is allowed on. And the patients will undergo either Adjuvant and eventually PSMA PET once that amendment goes through. And then they'll be broken down into two different randomizations based on whether extrapelvic metastases are found on PET or not. And for the group that does not have extrapelvic metastases, the question is about the enhanced systemic therapy with apalutamide. And for the patients that do have extrapelvic metastases, apalutamide is included for both arms. And the question is whether metastasis-directed therapy leads to better outcomes. Progression-free survival, as you can see, is the endpoint. And currently 25 sites are approved already and 13 patients are enrolled. So please keep this study in mind. And again, the key thing is PSMA PET is coming on the next amendment. Thanks.
All right, thanks very much, Evan. We just have this open at our site soon, so it's taken a while, but hopefully we'll get a contribution here. And erudite and to the point, Evan, as usual. All right, Paul Korn, if you can come back and give us the briefest of update on Charted 2.
Yeah, basically Charted 2 completed enrollment, so that's exciting. And I checked with Christos, who's the lead PI, and there's just no data update yet, so nothing else to report. But it's finished, and I think we played a good role in that.
All right, and well done to the folks at Kaiser Permanente. Helen Moon, Andrea Harstark and co in leading the accrual from SWOG on that one. Also, obviously from MD Anderson and Farmington Huntsman. Thanks. Okay. So, Dr. Dorf, eradicate. Is Dr. Dorf with us? I don't know. She was emailing earlier. So, this is
David, she's saying, Tanya is saying she doesn't have the privilege to speak here. She should be a panelist, but she's joining as she has joined as an attendee only.
Okay, Courtney or someone, or the IMS folks, can we help getting Dr. Dorf privileges to speak? And we will come back to her when she's able to speak to us. Okay, Abhishek Solanki is on. Hopefully Abhishek can speak to us about NRG GU005.
Yep, I think. Thanks for the intro. So this is GU005, and you can go on to the next slide. So it's a study led by NRG, and it's a randomization of a key question in radiation oncology, which is what is the role of SBRT for patients with localized disease? So it enrolls primarily favorable intermediate risk patients and randomizes between moderate hypofractionation using 60 or 70 gray and 20 to 28 fractions versus SBRT using 5 fractions. You can go to the next slide. The primary endpoint is actually a composite endpoint looking at the incidence of toxicity with treatment as well as disease control with multiple secondary endpoints, including a translational MRI study for patients who develop biochemical failure. And then you can go on to the next slide. So ultimately, it's accruing really well. So most recently, the 60 and 20 was allowed based on the CHIP and PROPHET studies. As of last month, it's very close to complete. Well, actually, the most recent numbers are 560 of 622. So it's anticipated that it'll close in January or February of next year, and hopefully we'll have some results in around 2024.
All right, so this is a good trial that's actually going pretty quickly, and it suggests there is an appetite for getting trials with SBRT done. So I think for other things that we propose might be a reasonable base. Do you agree with that?
Yeah, absolutely. I think that there's a real opportunity. I think it's very easy to put patients on this study because it cuts down visits from 20 to 28 treatments to 5 treatments. And it's still an unknown about the high risk patient population as to the role of SBRT for those patients. And I believe there's some studies that are in development in that setting as well. But absolutely.
Good. All right. Now, unless Dr. Dorf's ready to speak to us, I'm going to just keep going.
I think Dr. Dorf is here.
Okay, Dr. Dorf, are you with us?
Yes, I am.
Sorry about that.
Thank you.
We would never mute you.
The Eradicate study is struggling, so I'm glad to be able to speak to you just to ask you to look for these patients. It's for high-risk patients after prostatectomy. High risk isn't just defined by what you might think. It's similar to 9921 that we ran some years ago. So you start with a CAPRA score. So for instance, a Gleason 4+3 with a positive margin could potentially qualify. So think broadly about patients who might be eligible. They get a free Decipher test, and then they get randomized to a year of ADT or a year of ADT plus darolutamide intensification if they have a Decipher over 0.6. If they're under 0.6, they don't go on the study. So one of the struggles I've had is that most of my high-risk patients tend to have lymph node positivity, and lymph nodes are not allowed. But if you have had any other patients who screen failed, please email me or put in the chat so that I can give that feedback to Alicia Morgans. Thank you.
Yeah, I think this is just another example of studies that are slow to open. And I think we'll get there. We just need to keep pushing on it. And I am in trouble with Dr. Morgan. She said she wants to speak to me. So hopefully I won't get in too much trouble with her. All right, thank you very much. So we need to work on that one. And then quickly, GU007, David Vanderwill.
Great, thanks. So this study is looking at the question of PARP inhibitors, specifically niraparib, to sensitizing to radiation. And this is a phase 1/2 study. It's currently in the initial phase 1 portion, which takes a bit of time since we're watching patients through their radiation and looking for toxicity during that time point. So there's an initial sort of dose escalation, and it's currently at dose level 2 of niraparib with 200 milligrams of niraparib, except while they're getting radiation therapy, it's dropped down to 100 milligrams. And it's in the DLT period, observing during that time period before progressing on to dose level 3, which will be 200 milligrams throughout for 12 months. Currently, it's open for patients. I believe it's restricted to Gleason 9. Once it moves on to the phase 2 level, if you could, so dose level 3 is expected to open up again for slots in November, assuming that there are no DLTs. And then eventually, once the dose is established, we'll move on to phase 2, which is shown here, which will a little wider eligibility, including 7 and 8, and then patients will be randomized to IMRT, ADT with or without niraparib at the dose established in the phase 1 portion. Primary objective being PSA remaining less than 0.1 at the end of ADT therapy.
All right, thank you very much, David. And just as we move forward, just to be clear, it's agnostic for DDR, right?
Correct.
Okay, thank you. All right, Dan Lin.
Oh yeah, this is continuing on with the NRG trials. This is GU008, so-called Innovate. So this is a node positive group. You can go to the next schema. Kind of takes the what Tanya was saying. This is the node positive people. So as Eradicate does not accept them. This is the trial for node positive. We talked about this at various other meetings. So node positive after prostatectomy, and then they're randomized. You can see, go back. They both get RT. They're RT with or without. At the trial was designed for ABI and APA, and they all importantly get a PSMA PET, or I'm sorry, a PSMA or FABC PET at the entry or choline. And the primary endpoint was MFS. So there have been some modifications. They have about 45 patients, I think, enrolled nationally. And then the next slide will just show you the modifications that are underway. Some amendments, basically little things of logistics, duration of ADT. They're having some combination issues, and then they're going to change that from ABI-APA to just APA. So they're kind of on hold now. I just talked to Ron Chen. The target's over 550, so they're just early. And I think, as Dave said, this is another one like Eradicate, sort of slow to enroll and slow to open up. But I think it is an important node positive trial. Really, the only one out there as an unmet need amongst all the NCTN trials.
Okay, thank you. And then finally, GU009.
Jim is seeing patients. This is the same NRG trial, still on hold for amendment, same issues, GUA, but it's moving along. It will be reviewed at Central RB next week. Hopefully it will be open very soon. More to come.
Okay, all right. And we just note that there. All right, thanks everybody for really moving along today. I think we did get finished just a little bit after 1:30, and I'm going to hand back to Ian Thompson to close us out.
I think it's perfect timing. I'd like to thank everybody. It was a great meeting. From everything that we can see, all the indicators would suggest that come spring, we will be back together again. So until then, any last minute items to bring before the group before we close and go on to a great weekend?
Yeah, I just have a quick announcement. So on this call, we had one of our community advocates, Joel Poynton. He's a prostate survivor. And I just want people to be aware we have a community advocate program, and its intent is to bring some of the underrepresented communities, give them a voice. So Joel joined us today, but we have a number of other advocates in that space. There are 10 open slots, and we have 6 of them filled.
That's great.
Rick, can you send me Joel's contact details? I have to go, but I'm very much keen to get Joel involved in our prostate work. Thanks.
Okay, thank you.
Excellent. All right. Everyone, thanks so much to the leadership and to those who all presented today and yesterday, and let's go forth and do good work. Blessings to all. Take care. Great job.